types of inoculants. And what is an inoculant? Well, in the traditional sense, an inoculant will be associated with soybeans, or you would think of a rhizobium product, but in the correct definition, inoculants are bacteria or a fungi, a living organism that improves plant growth either by freeing nutrients for plant use or entering into a symbiotic relationship with the plant, or a good marriage, not a bad marriage, a good marriage, because there are relationships out there that are bad marriages, and those are called parasites. And then acting as a biocontrol against plant pathogens. There's several different organisms in the marketplace today that act against Pythium, Rhizoctonium, Fusarium, and they are actually living organisms. Traditional inoculants in the sense that we're using it on a legume, and that refers to soybeans, alfalfa, dry beans, clovers. This was where we would think of traditional inoculant use. And inoculants with these type of organisms, these type of plants that were actually taking nitrogen out of the air and giving it to the plant, in turn the plant is giving the bacteria a place to live. So these bacteria live within a specialized structure called a root nodule, and it's a place where they can process atmospheric nitrogen and give it to available form to the plant. Nitrogen is a much needed nutrient in crop production, especially soybeans have a tremendous need for nitrogen, and we'll talk about that a little bit later. We'll talk about non-traditional inoculants, and these will be products that not so much rhizobium based or used with soybeans, alfalfa, peas, or beans. These organisms do not live within the specialized nodule that you would find on a soybean plant. These organisms are not limited to, I'll just mention a few. Basically, you have bacteria, and the main ones that are used in the industry today are Bacillus species and Azospirillum. And then we use also beneficial fungi. The trichoderma and then penicillium are also used in the industry today. So why would we want to use an inoculant? Well, basically microbiology is cheap labor. And it's on the payroll, we don't have to pay taxes on them. And what do they do for us? Well, they provide season-long response for crop nutrients such as phosphate and nitrogen. We're going to talk about the nitrogen demands for soybeans a little bit later. But these microbes, they can free up phosphate, they can free up other nutrients such as nitrogen. They actually can suppress or even control diseases such as Pythium, Rhizoctonium, Fusarium, season long. A lot of times when we think about disease control, we've actually identified an active ingredient, supply an active ingredient to the plant that has a very short half-life. Microbes can live with the plant the entire life cycle of the plant and control the pathogen. Then also we get a crop stimulation or a PGR type of effect when we're using an inoculant product. We actually can control the expression of the genome of the plant. So certain microbes, if you inoculate the root, it actually does control the expression of, say, how much leaf matter you get above ground. So this is kind of a global summary of trichoderma. Trichoderma are beneficial fungi. And this is really a complicated type of chart, but it just kind of shows that microbes are very complicated in the way they interact with the plant. Bottom line, they do change plant physiology. By changing plant physiology, we have more roots, a larger diameter stalk, and more leaf matter above ground. That means we can extract more nutrients, we have a plant that stands better, and we can harvest more sunlight. So for example, the true mode of action with trichoderma, and this research is coming directly from Cornell University over in New York. We know that when the beneficial colonizing fungi down at the bottom there interact with the plant root, it actually causes a cascade of events. And the true mode of action, we're actually affecting the jasmatic and ethylene signaling pathways in the plant. So what's that mean? We're actually turning on genes, and that gene actually induces physiological response in the plant. So that's really a technical kind of explanation. I know if I was talking about microbiology, say 5-10 years ago, I would think I would get lumped into a group of people that would maybe not advocate paying their taxes or be kind of thought of as a kook. But you know, if you look at traditional agriculture 10, 15 years ago compared to where we are today, we have products in the marketplace like Poncho Votivo, other products that are actively using microbes. And we know that microbes can influence crop physiology. So if you look at this is some of the work that I've done personally back in 2007 in the selection of certain strains of fungi. We've got the K2, K4 combination. So we can see a control plant, see a K2 where the above ground portion is actually larger, the K4 where the below ground portion is influenced, and then when we put the two species of beneficial fungi together, we actually have a response both above and below ground, just through the colonization of the root with a fungi. So what we also see, we see increased root expression with and without here. We also do see effects on seedling germination. This would be with and without. The two rows to the outside there are treated with a biological product, and the rest of the field is not. We do see above ground responses. This is winter wheat, treated with a beneficial fungi, work done over by Cornell. And you can see that we have more tillering, more root mass, and more heads per plant. Biology cannot replace a complete sound nutrient program. They're not meant to replace your MP and K programs. They're there to complement it. You also need to have an integrated crop disease and pest controls in place. And one of the things that I'm a strong advocate of, if the plant is sick, all this biological stuff is not going to work well. So if you have root rot, and if you let root rot get out of hand, there's no way, for example, that you can have nodulation occur. You have to have a healthy plant, or give the plant at least the opportunity to be healthy. And I think our previous speaker spoke a lot about using some of the actives as far as seed treatments to present some of the early diseases, Pythium, Rhizoctonium, Fusarium in soybeans. And they're not a silver bullet, and this point's been going over a few times. They can't cure everything from a broken bone to a broken heart. They are meant to specifically address certain problems in crop production. I want to focus the rest of my presentation on soybean and soybean production and inoculants. One of the things that I always like to refer to when we're thinking about soybean production is the components of yield. How many gallons of water does it take to produce a bushel of beans? 13,500 gallons of water. We'll look at nitrogen. How many pounds of nitrogen per bushel? 5.5. So if I'm going to grow a 60 bushel bean crop, how many pounds of nitrogen do I need? A lot. Over 300 units. So you know, you look at P, you look at K, you look at the pounds of sugar that are needed to produce a bushel of beans. Well, over 300 pounds of glucose, or about 4,000 semi-loads of air to extract the carbon dioxide to produce the glucose for the plant. So soybeans in general are a tremendous feeder of all nutrients, and especially nitrogen. And this is where the relationship between microbiology and the soybean plant really comes into play. So if the soybean plant requires 5.5 pounds of nitrogen per bushel grain and stover, how much nitrogen did you apply to your soybean crop last year? Is there anyone in here that actually applied nitrogen to the soybean crop? And just very little. I mean, generally, we're not buying. Is anyone buying urea to put on the? And this is something where, if we didn't have the relationship between the microbes and the soybean plant, I don't think anyone in this room would be willing to spread 700 pounds of urea to make up that difference. Because that's what the microbes do. So when I talk about microbes being cheap labor, 700 pounds, you think about the bean's protein packed, that protein is mainly nitrogen. It's coming from someplace, and it's between the relationship of the plant and the microbes. So nitrogen is an important aspect of producing high yield beans. I don't know if there's many people in this room who have actually seen a field of beans that haven't nodulated. Typically in the area that we are today, there's been a lot of beans grown for many years, but Occasionally you do run into the areas where either it was in corn for a long time or coming out of CRP, or we've had some adverse weather conditions. And I put this slide up here just to show the difference in vigor. And these are young beans, we're talking V2, V3. Even at this stage, you can see the difference with and without root nodulation, the difference in vigor and the overall health of the soybean. And that goes back to the direct relationship of the amount of nitrogen needed to produce a bean. And the beans there too, on the other side that don't look as healthy, they're just relying on any nitrogen that would be left in the soil or mineralization of organic matter. So why did we stop inoculating? Well, as farms became bigger, and you talked to a lot of the older guys, and everyone can kind of remember when they first started planting beans, and they all knew to use an inoculant. And you took the bag of dirt and you dumped it in the back of the drill, and then you got the axe handle or the canoe paddle out, stirred it around. You planted your beans, you opened up the drill box, and all the dirt was still there, and every once in a while you had a plug tube, and it really didn't stick very well. So I think as farms became bigger, inoculation wasn't that convenient to do, and something that was just often overlooked just because the production practices and the delivery methods really hadn't caught up to today's farming. So what's different now as we look ahead? Well, the industry as a whole, as far as rhizobium production or inoculation for soybeans, really has changed in the last 10 years. So when you look at what the industry has done, we've really gone to more sterile packaging. We have changed our genetics. There's actually genetics out there today that are brand new. When's the last time you've heard about a new strain of rhizobium introduced into the, there is new strains out there. We're not relying on, say, going to the USDA data bank and buying a slant. We're actually matching up the genetics to today's commonly grown genetics as far as soybean varieties. And then also, they are a lot easier to use, no more getting the canoe paddle out and stirring it in. Now, if I would put this up here and go back to our Agronomy 101 days, everyone would agree with me this is probably excellent nodulation. And this concept is absolutely false today. This is considered very parasitic and inefficient when we look at today's current inoculant strains. So this would be the new super strains that are in the marketplace today, and there's actually a physical difference when you look at the nodule itself. So if we look at that picture and then this picture, the first thing that strikes me is the size of the nodule. Then also we look at the size. So the newer genetics have a larger nodule. We actually have less nodules per plant, so we have less mouths to feed. And then what we have is this wrinkle type of appearance. We call that crundulation. So the nodule itself has a larger surface area for gas exchange. And they're anywhere from 30 to 100% more efficient than even strains that we had 10 to 15 years ago. So then when it comes to nodulation, there is a difference in genetics. And I think this is something that everyone needs to be aware of, that the soybean inoculant that we had 15 years ago is not the same soybean inoculant in the marketplace today. So we can see improvements, especially in rotated ground. So why are we seeing yield increases in rotated ground when we haven't in the past? Well, we do have better strains of rhizobium, so it comes down to genetics. And we do have better production methods and delivery methods. So when it comes to actual yield coming from that, coverage is key with delivering inoculants. And then also some of the things that we've been able to do is really figure out a way to pack super strains together without them fighting with each other. So the new soybean inoculant formulations offer a variety of things that we didn't have 10 to 12 years ago. First of all, we have higher cell counts and higher concentrations. Why is that important? It's important because if we look at the number of cells or the number of rhizobium per plant, there's actually only about 200 infection sites on the plant. We want to crowd out the native bacteria, so we start with about 1 million to 1.5 million cells when we treat it. With the most traditional products, you treat it, within 2 hours you're down to 200,000 cells, but you're still at 1,000x of what you actually need to crowd out the background population. The other thing that's really important today is that there are products in the marketplace that actually combine multiple strains of rhizobium. So we have multiple strains co-packaged without inter-package competition. What does that mean? That means if you put two bullies on a school bus, they usually fight with each other and someone comes out with a black eye. With some of the newer technology, we're able to package super strains together and deliver super strain A, B, and C to your farm without them fighting with each other. So you don't end up with just one dominant strain in the package. And the other thing is you can pre-treat inoculants now several months ahead of time. One of the new innovations for grower-applied inoculants would be the ability to get away from the peat or humus application into the planter. You know, most planters today are $100, $200, $300,000 pieces of equipment, and you know, pouring dirt into them is really not a good practice. The last time I bought a radial beam meter, it was $800. And you know, if you talk to Kinsey, they say that peat or humus-based products really are detrimental to that. They tear them up. So there has been advancements in the marketplace that we've actually been able to take talc and graphite combinations, blend that with rhizobia, and being able to use that into a carrier. So no longer do you have to dump dirt in the planter for an inoculant. You're dumping talc and graphite that has the rhizobium. So, you know, things have really changed here in the last 5 years, especially for stuff that you want to apply yourself. And it is easier to apply, and the coverage is greatly improved over the dirt inoculants of the past. The other thing that has improved, I've been treating seed for a long time, well over 10 years, and I've visited a lot of seed treatment sites. And there's always been one common, or two common pieces of equipment there when I show up. One's a rubber mallet or hammer. The other one's a broomstick that's either blue or red on the end of it. Because a lot of times treating seed, it's gooey, it's sticky, and things bind up. And no one wants to go pick up a wagon load of beans, lift up the chute, and nothing comes out. So inoculants, and rightly so, over the last few years have gotten a really bad reputation of causing sticky problems. There are improved liquid applications in the industry now that do eliminate the sticky problems. I want to cover really quickly here inoculant fail factors or how not to throw your money away when buying a biological product. These are things that you guys need to be aware of if you're going to use an inoculant. These are things you do not want to do unless you want to throw your money away and have a dead product on your seed. The first thing is storage. We need to make sure we are reading labels and storing the product in proper conditions. That means cool and dry and out of the sunlight. We wouldn't buy a gallon of milk and put it in the pickup truck, roll up the windows and let it sit there for a week and expect to drink it afterwards. It's not a good thing, and the inoculants are the same way. The other thing is water. When you look at, I'll say, a seed treatment, or you're adding additional water to seed treatments or using water on soybeans in any way, shape, or form, it better not be chlorinated. Or if it's chlorinated, you need to make sure that you've removed all the chlorine out of it. I'll share a story with you. A large company actually built a new seed treatment unit in town. They had chlorinated water. They had a bulk treater there. They knew they had to remove the chlorine from the water, so they were using a dechlorinator. Well, they ran out of the dechlorinator. They sent Bubba John down to the hardware store to pick up more dechlorinator and treated the beans, and then Vince got the call, hey, we've got 40,000 acres of beans down here, there's no nodulation. You need to come look and see why your product's not working. So we go through the typical, you know, pain of talking to growers and then explaining how they're going to lose money and looking at fields that look really bad, and then we go back to the plant. We talk about the water. Well, they knew they had chlorinated water, and they knew they had to use a dechlorinator. And I said, well, can I see your dechlorinator, please? It said, yeah, it said pond dechlorinator plus bacterial side. So, I mean, you know, that's a situation that's real life that affected a lot of acres of beans. So my suggestion, if you guys do have bulk treatment sites or are using any type of chlorinated water, what I like to do is go ahead and work with our local Culligan or water place, and the spigot that's closest to the seed treater, put a dechlorination system on it. That way, that same guy that's pushing the broom is usually the same guy that's treating seed a lot of times, so he can just open up the spigot and not forget to put it in or go down to the hardware store and buy the wrong product. The other thing is, how are your products handled after treating? A lot of the inoculants, when treated the proper way, are good products and they're alive. It's just what happens to them afterwards. Are they put in a semi-truck and it goes down the road and it's 85 degrees out? What's the temperature inside the truck? Are you putting your pro boxes out in the sun and letting them sit there and cook? We actually did a study down in the south in June. What do you think the temperature inside a central fill unit is? Well over 120 degrees. So, you know, those are some of the things that you've got to evaluate when using inoculants, especially on ground that isn't, that really needs it. The wrong type of product, soybean inoculant will not nodulate alfalfa, or alfalfa inoculant will not nodulate soybeans. So making sure that you have the right rhizobium to get the job done. Poor coverage. Coverage is key, just like in spraying. If you don't have the inoculant on the seed, it can't get the work done. The other thing is expired product. Inoculants do have an expiration date. It's not like going and buying a 2.5 gallon jug of Buctrol. You can keep that for 5 years and it's still going to work. Inoculants do have an expiration date. Some of them are 1 year, some of them are 2 years. So you need to make sure that you're checking expiration dates and making sure you have fresh product if you're going to use a biological application. And then lastly, improper planting practices. What's the correct planting depth or the average planting depth of soybeans? An inch. Sean Conley up in Wisconsin, we'll pick on the Wisconsin guys a little bit, did a survey. 78% of his respondents came back 2.5 inches deep. So we know exactly what they're doing. They're not changing their planter after planting corn. So planting too deep can be deleterious to inoculants. The other thing I want to touch on is pH. If you have a pH of less than 5.5, 5.4, you're probably not going to have that many nodules. Nodulation is greatly affected by pH. It's also affected by drought or flooding. You know, a lot of guys think drought affects it. It's not so much the water aspect, it's because it's become anaerobic. Rhizobium need oxygen to live. A lot of the inoculant products today do come in a liquid. So it's not that it's the water that hurts them, it's the lack of oxygen. And then also, the other thing I want to touch on is soybean history. For every year that you're out of soybeans, you lose about 1/3 of your background population of rhizobium. So that's why we say a lot of time in the industry, 3 years corn, it's a really good, really good idea to be using an inoculant with your soybeans. And even in the corn-soybean rotation, with some of our more challenging soils, we do see a response today where we didn't see a response, say, 5 to 10 years ago with the older production methods and delivery methods. So in summary, we're talking about biological inoculants, the next generation products. They do increase efficacy for corn, wheat, and soybeans. So not only are inoculants thought of now today just for soybeans, you can use them on corn and wheat, as we talked about. The combination of strains that really have been niched against the combination of species and current genetics for optimal results. What's that mean? Are the genetics of soybeans the same varieties that were growing in 1997? No. A lot of those ISO lines have gone to the wayside. New ISO lines have come on board. Various soybean varieties have different marriage patterns, and what we've actually done in the industry is try to match the genetics today to our current line of soybeans that we're growing. And then also, there is greatly improved formulations. The days of dumping dirt in your planter are done, guys. You can actually dump something in there that's going to improve flow. You know, treated seed always needs a lubricant. I mean, you're going to need that. Most planter manufacturers do recommend a lubricant. And then also, lastly, we do see improved ROIs with the new inoculant formulations. You know, look at where a bushel of beans is today versus in 2004. You know, $13 versus $4. So our ROI numbers do get a lot wider with some of these products. Does anyone have any questions, comments? Anything I can talk about as far as inoculation, seed treatment? that anyone may have. Yes?
Does it have to be on the seed, or can you put it in with your broadcasting or pre-emergent herbicide, spray it on so it covers the whole ground?
The most economical method of inoculating soybeans is to have it on the seed. And you can broadcast or go in furrow, but it's going to cost a lot more money. The reason I say that is because the relationship between the rhizobium or the inoculant occurs 3 days after the radicle emerges. So it occurs really early in the life cycle of the plant. So seed applied is always usually the most effective and the most cost efficient for you as the grower to use. But we do have guys that a lot of times will want to run it in furrow sometimes, just out of ease. They don't, they feel that mixing the product in the planter box is not that effective. Do you have a question?
Yes, I just wanted to ask, because it's been quite a few years since it's used, and usually our routine out of continuous corn back to soybeans. In the past, we were trying to mix it in the seed hopper. So I didn't know if there's a product that we would recommend that would work better for new planters.
The question was regarding coming out of continuous corn and using a on-farm product as far as a recommendation. I think if you were to look at something that has more of a talc graphite or a lubricant aspect, I do work for ABM and we do have those products available. But if you look at, a lot of guys today are using seed lubricants when you plant soybeans. So my response to how do you apply it, the first question I ask, how do you apply your seed lubricant? And and there's some equipment out there today. I think one piece of equipment is made by EnviroPack up in Compton, Illinois. It's a little dry applicator. If you have a buggy or a bulk system, you can actually be applying the talc graphite mix right into the seed stream as it's going up the brush auger. So the process becomes very automated and hands-off, not like the days of dumping bags and stirring it in. So some of the on-farm application, we all have to use talc and graphite. So why not use a talc graphite-based material that already has all the biological in it? Any other questions? Well, I do appreciate your guys' time. We do have a rep here in Illinois, Bill Winter. So if you guys, Bill's right here. If you guys have any additional questions, you can always contact Bill or myself regarding inoculants and biologicals. And thank you for your time.